Okay, so today I'm going to be talking about mitosis as part of the cell cycle. Okay, so the cell cycle is divided into interphase and mitosis. Mitosis is only about 5 to 10% of the actual cell cycle. Interphase itself is subdivided into two gap phases, known as G's phases, and a synthesis phase, which is the S phase. Okay, so the first gap phase, which is also known as G1, during that, proteins are synthesized, organelles replicate, more new cytoplasm is made, and at this point it's past the G1 checkpoint. And after that, the cell is cleared to move into S phase. Okay, so S phase, the synthesis phase, DNA replicates by semi-conservative replication. This is where each molecule of DNA unwinds and unzips, and the hydrogen bonds between complementary base pairs break. Three nucleotides pair up to the exposed nucleotide bases on the DNA strands. So there is complementary base pairing between A and T. They both pair with 2 hydrogen bonds, and G pairs with C by 3 hydrogen bonds. This is catalyzed by DNA polymerase, which is an enzyme. Then DNA ligase catalyzes the condensation reaction between a sugar group and a phosphate group of adjacent nucleotides. By the end of the S phase, each molecule of DNA has been copied into 2 identical molecules. This cell checks that these are identical to the original molecule, and these become the sister chromatids. Okay, then we move on to G2, which is the second gap phase. Cells and organelles reach full size. Synthesis continues here, and the cell builds up energy reserves. The centriole divides into 2. This passes what is known as the G2 checkpoint, and cells are cleared to enter mitosis. Okay, so mitosis itself is split up into several stages. After interphase, we have the first stage, which is prophase. During prophase, the chromatin, the DNA of chromosomes, is condensed and tidally coiled into chromosomes. Because the DNA is replicated during interphase, each visible chromosome consists of 2 identical sister chromatids joined by a centromere. The nuclear envelope breaks up at this point and disperses into the cell, and the 2 centrioles migrate to opposite ends of the poles. This makes the 2 poles. We then move on to metaphase. During metaphase, a spindle made from microtubules form, and chromosomes, which the pairs of sister chromatids, attached to the equator of the spindle by the centromeres. We then move into anaphase, and at this point, spindle threads shorten, causing each centromere to break, separating each chromatid from its sister. As you can see, they're pulled apart. And the separated chromatids, which are now called the chromosomes because they're separate and individual, they're pulled to opposite poles. So this then moves into telophase. So a nuclear envelope forms around each group, known as double set, of chromosomes at either end of the cell. The chromosomes uncoil, and there are now 2 diploid nuclei, each genetically identical to each other, and the nucleus of the parent cell. The cell now undergoes cytokinesis, where microtubules form a drawstring just inside the plasma membrane, and this pinches inwards and fuses. So now we have 2 completely identical cells.